Tissue perfusion is mainly a function of arterial blood pressure. In addition, it is also regulated by local factors within each individual tissue. This process is called autoregulation or self-regulation. Autoregulation ensures that the correct amount of blood is delivered to match a tissue's activity level. It acts on the smooth muscles of the arterioles and the precapillary sphincters. There are two main types of autoregulatory controls. The first type of control are metabolic controls, which are in response to the metabolic activity of the tissue. When tissues are metabolically active, they are using oxygen. Thus, the oxygen level in the blood is low, since the oxygen is leaving the blood and going into the cells. During aerobic cellular respiration, or the synthesis of ATP using oxygen, carbon dioxide is produced. Therefore, in metabolically active tissues, carbon dioxide levels will be elevated. Recall that carbon dioxide reacts with water to produce carbonic acid, which then dissociates into hydrogen ions and bicarbonate ions. An increase in hydrogen ions will also decrease the pH. So let's look at what will trigger the precapillary sphincters to open and the arterioles to vasodilate. It will be decreased oxygen, increased hydrogen ions, and an increase in carbon dioxide levels. Again, all of these are due to increased metabolic activity. So metabolically active tissues will receive more blood to provide the oxygen required and to remove the waste products that are being produced through aerobic cellular respiration. Flow to a tissue when it is increased above normal or elevated is called hyperemia. The second control mechanism is the myogenic mechanism. When smooth muscles are stretched, such as in an increase in blood volume, vasoconstriction occurs. So smooth muscles naturally contract when they are stretched. The opposite is true. Decreased stretch leads to vasodilation. This myogenic control mechanism regulates blood flow into the true capillaries. Capillary blood volume and thus blood pressure will influence the exchange of materials between the blood and the tissues.